All right, in this video, we're going to talk about DNA repair. So there are several ways in which DNA can become damaged, and that damage needs to be fixed before it becomes a mutation. There are errors in replication that can occur. There's various spontaneous damage that can occur, like deamination and depurination. And there's all sorts of chemical and environmental damage that can occur. So, for example, UV light can damage DNA. Now, what we need to first discuss is the difference between DNA damage, or sometimes called a lesion in DNA, and mutations. They are different. Okay, a mutation is not just another word for DNA damage. So let's illustrate what I mean by the difference of these two. So let's say we have a double-stranded DNA here, and the cell in which this DNA resides is going to divide. Therefore, the DNA needs to be replicated. So this strand here is going to be the template, and so the daughter that gets made is right here. And you can see that DNA replication occurred normally and everything looks good. Here, on the other hand, so this strand is going to be the template, and the daughter strand that gets made is this strand up here. But notice, there's a mismatch. Instead of putting an A here, a G has gone there. Okay, so now this is abnormal. The cell can detect, hey, wait a minute, G's are not supposed to pair with T's. So this is considered DNA damage. This is considered a lesion because the cell can detect that there is a problem here and the problem needs to be fixed. Because if it's not fixed in time, here's what can happen. This strand here, which is still our normal strand, this strand here is going to act as a template. If that cell, so if this cell here in which this DNA resides is going to divide again, then if this strand here, parental strand, acts as a template in which this strand, newly strand, synthesizes, everything is normal. There should be a T here. An A was properly paired. Beautiful, wonderful. This is identical to grandma over here. However, this strand here, okay, now if this strand here, if it is allowed to be used as a template before this G is fixed, then what happens is it is read, that G is read, and a C is put there. And now we have a normal GC base pair. Now we have a difference from the grandmother here. If there was an AT, now it's a GC. G's are supposed to pair with C's. This is now a mutation. The cell cannot detect that this is not supposed to be a GC base pair, and therefore the cell cannot fix this, and this is a permanent change in the DNA. It is a mutation. So the trick here for cells is to repair DNA while it's damaged, while it's a lesion, while it can detect the problem before the problem becomes a mutation. This is important, right? You don't want changes occurring too often within DNA, and so there are all sorts of repair systems. In fact, there are about 130 genes in humans have evolved to repair DNA. In fact, mutations that knock out any of these genes or disrupt any of these genes are going to cause problems for cells to repair their DNA, which means more mutations are going to arise, which means those cells are going to be more likely to develop cancer. Okay? So these repair systems are really important. There are lots of different repair systems, and we're not going to cover all of them in this lecture, in this class. What we're going to cover is mismatch repair, direct repair, base excision repair, and nucleotide excision repair. So let's take a look at some of these, starting with mismatch repair system in bacteria, in E. coli. Well, here we look and we see a cytosine. This is the nitrogenous base of cytosine. And now, like many chemicals, this cytosine can exist in different tautomeric states where you're sort of pushing electrons around. This is the primary tautomeric state of cytosine, and this is the state at which it pairs best with guanine. However, there is a rare tautomeric state, right, where it no longer has this hydrogen here. These electrons are over here in the double bond here. Well, now we don't have a hydrogen donor here involved in that third base pair with the guanine. And so this cytosine in this a rare tautomeric state can sometimes mismatch and cause mismatches. So DNA is double-stranded. Okay, so if there's something messed up here with X, then all the cell has to do is go, okay, well, we know what should be here, because if there's a G there, there should be a C here. So it removes that X and it pops in a C. Beautiful. However, what happens if the mismatch is like this? Uh-oh. Let's see, there's definitely a mismatch here. We're not supposed to pair a G with a T, but are we supposed to read the G to put a C here? Or are we supposed to read the T and put an A here? Right? How does the cell know which way to fix this problem? And the answer is, what the cell assumes is that the parental strand is always correct. That if there is a mismatch, the cell will look at the parental strand and use that as a guide to fit the more recently replicated strand. So if it came across something like this, it would read that C in the parental strand and fix this T into a C. Well, the next question is, how does a cell know which is the parental strand and which is the recently replicated strand? And the answer is methylation. There is an enzyme called DAM methylase, and what that does is it methylates adenines that are specifically part of this sequence. Every GATC, bang, that A is methylated. Every GATC, bang, that A is methylated. Now that DAM methylase doesn't work immediately. So when replication occurs, there will be a certain amount of time in which the DNA is hemimethylated. At each of these GATCs, only the parental strand is methylated. The newly replicated strand is not methylated. Now that doesn't last forever. Eventually that DAM methylase kicks in and methylates even the newly replicated strand. So there are a few minutes, there is some time in which the DNA, the cell can recognize which is the parental strand and which is the newly replicated strand, that they are labeled. So it's in this period of time that any mismatches that have occurred within the DNA need to be fixed now. How does that work? Well, there is a protein complex called mut-L and mut-S. And mut-L and mut-S form a complex, and they recognize mismatches. They recognize this abnormal structure and they bind it. And then they start feeding through both ways the DNA until they come to the first hemimethylated site. And the first hemimethylated site, they then make a cut, and that is mut-H. It recognizes that hemimethylated site. It knows, okay, the A is methylated here, it's not methylated here, and it cuts on the non-methylated part. So now we have a nick in the newly replicated DNA that is fairly close to. So the next step is to eat back this DNA until we remove this all the way up to this mismatched base. Now, depending upon whether that nick had occurred upstream of the mismatch or downstream of the mismatch will depend upon what exonucleases are used. Okay? One way or the other, there are going to be a series of exonucleases that chew all the way to the mismatch. Chew all the way to the mismatch. And then after that's done, the rest is very straightforward. We bring in DNA polymerase 3, which recognizes this gap here, and it just starts adding. Add, read, write, read, write, read, write, read, write. And then all the way at the end, we get a DNA ligase that then ligates this fragment to this fragment, and we're done. But what this means is that in order to fix a single base pair, a single mismatched base, the cell is willing to destroy up to about 500 base pairs of single-stranded DNA simply to fix the one mismatch. And that demonstrates just how important it is to keep the fidelity of, or the accuracy of DNA replication intact. All right, next, the repair of DNA damage, so of unnatural bases or disruptions of DNA structure. And the first is these DNA photolyases, and this is direct repair. Instead of cutting out a bunch of DNA, we're going to try to directly repair this. We've got UV-induced thymine dimers. So here we have two adjacent thymines. If they absorb enough UV light, what happens is a covalent bond occurs between the two of them. This is called a thymine dimer, and it causes a distortion in the DNA such that if replication were to try to occur to this DNA before it's repaired, that could cause deletions and insertions and all kinds of problems. So there are enzymes called DNA photolyases. And what a photolyase does is it has the ability to recognize these thymine dimers because they cause this sort of irregular structure within the DNA. Now this reaction here to get rid of that covalent bond is a fairly endergonic reaction. It's going to take energy to break. And so that photolyase actually couples the energy from light in order to fix this, right? To directly break that covalent bond that has occurred between those two thymines. And now we've repaired it and DNA replication can occur unhindered. Another example of just fixing a single base without chopping out a whole bunch of stuff is referred to as base excision repair. And what this fixes is oxidative deamination. So there is a particular reaction that can occur to nitrogenous bases that is spontaneous. And that is replacing an amino group with a carbonyl. This is called oxidative deamination. And what happens is when this happens to a cytosine, it becomes a uracil, which is then going to pair with an adenine. If it happens to adenine, it becomes hypoxanthine, which pairs with cytosine. If it happens to a guanine, it becomes xanthine, and now that only has two hydrogen bonds with cytosine, which can again cause some mismatches to occur. So this oxidative deamination has to be fixed. Otherwise, right, there's going to be a problem. We're going to get mismatches. Well, hypoxanthine is not a normal base, so boom, that can be fixed. That can be recognized. Xanthine, not a normal base. That can be recognized and fixed. But look at this. C to a uracil. Well, right, uracil is a normal base in RNA. And in fact, it's believed that it was RNA that evolved first, and then DNA evolved from RNA. So the hypothesis here is that the reason that DNA evolved thymine to replace uracil is because of this here, because of this ability, this spontaneous mistake that can cause cytosine to become uracil. If DNA maintained uracil instead of thymine, then when cytosine becomes uracil accidentally, the cell would have no way of knowing that that is not supposed to be uracil. So through evolution, thymine replaced uracil in DNA, and now when cytosine becomes uracil within DNA, the cell can tell, wait a minute, that's wrong. Uracil isn't supposed to be in DNA. Let's fix that. So it's kind of an interesting little chemical note there. All right, there is an enzyme called uracil glycosylase, and its job is to repair these cytosines that have become uracils. So the cell has that glycosylase. It recognizes uracils that are in DNA because that's not normal. It will then cut, boop, first it cuts off the entire uracil nitrogenous base, boop, there it goes. And now what we have is called an AP site. That means that we have a ribose there, but we don't have a nitrogenous base on it. And that is recognized by very specific endo and exonucleases. This AP endonuclease cleaves this bond. We break out this single base that is then replaced with a DNA polymerase, and things are ligated back together. Next, nucleotide excision repair. So here we have a distortion. Okay, now that distortion can represent several different types of damage, but that damage is recognized by a specific set of complex of proteins, UVRA and UVRB. They recognize it, okay, and then the strands separate. So A helps recognize, falls off. B, UVRB, separates the two strands of DNA. In comes UVRC, and now what C does is it cuts that single strand on both sides of the disruption there. Okay, it then is pulled off by UVRD, and then we get to our familiar territory here. We've got a gap. We've removed a hunk of one strand of DNA, this whole gap here. And DNA polymerase comes in. It adds to the end here, read, write, read, write, read, write, and then DNA ligase ligates that nick right there. So this is another example where the cell is willing to spend a lot of energy, right, to repair just a single little piece of damage. It will remove 100 base pairs or so, bases of DNA, just to fix that one piece of damage. Okay, so that ends our discussion of DNA replication and ends the material that will be covered on exam 2.